Okay, so welcome here to my introductory genetics lecture. So today I'm going to start off with asking the question, what is a genome? So there are many theories of what a genome is, but the one that I'm going to propose to you today is as follows. So a genome is DNA or RNA set of genetic instructions for an organism that can be replicated. So moving on with this term for genome, let's look at how genomes started in evolutionary history. So there are three possible ways that scientists have figured that genomes could have possibly started. So this is the protein beginning, DNA beginning, and RNA beginning. So first I'm going to take a look at protein. So proteins are capable of catalyzing reactions. They're able to alter other proteins. However, they're unstable. They don't hold conformation. They don't hold their shape. They degrade quickly, and there's no way for it to be a form of heredity. Now DNA is stable. It's actually very, very stable, which can be a bit of an issue. And it is not also capable of catalyzing reactions, which is also an issue. And thirdly here is RNA. And RNA can store genetic information and catalyze reactions. So scientists have proposed that RNA is actually the beginning of genomes. So now that we've figured out we have likely gotten to an RNA beginning here, let's look at how RNA transfers to protein and DNA. So this is a common figure that I'm sure most people have seen. But let's think if we started with an RNA beginning, then DNA didn't just strictly go to RNA, but RNA was also able to go to DNA, and we know this to be true. RNA then evolved a way to create proteins and DNA, and the cell then found that proteins are actually more efficient at catalyzing reactions, and found that DNA is actually more efficient at holding genetic information, and therefore DNA and proteins were likely born. So, genomes are very similar between all organisms. Because if we had an RNA beginning, and we have a common ancestor, then similar genetic systems among all organisms must be true. This simply means that research findings on one organism can be used as information for genes in other organisms. So, this is the family tree of where all organisms came about from Luca, our lowest common ancestor. But I have a question here. What happened at this point here? Because if you consider bacteria and archaea are kind of simple organisms, not in a genome-related way, but simplicity as in they're prokaryotes, and eukaryotes are much more extravagant organisms as they have many cells. So what happened at this location to allow eukaryotes to be so special? Well, the answer is eukaryotogenesis. This is endosymbiosis between bacterium and a prokaryotic host that brought this to be. So how does endosymbiosis occur? So we have cell 1 and we have cell 2. Now, one cell engulfs the other cell. So in this case, we'll say cell 1 engulfs cell 2. Cell 2 may provide some sort of advantage to cell 1, and there may become some sort of symbiotic relationship. And therefore, cell 1 is okay with keeping cell 2 within it. And over millions of years, this relationship is conserved, and cells begin creating less of what the other cell provides. Now we must realize that the ingesting cell does not digest the other cell, which is a very important component needed for endosymbiosis to work. So at this point, it's either parasitic or symbiotic. Both cells can still live without each other. But as more time progresses, more mutations arise, and more reliance on the other cell begins to occur. Also notice that mutations that may prevent one cell from living on its own do not prevent it from being successful, because it can stay within cell 1. At this point, cell 2 becomes trapped in cell 1, and an evolutionary ratchet occurs. Cell 2 is then stuck and cannot retreat from cell 1. Now that cell 2 is trapped in cell 1, it must stay and become more dependent, and will share more resources. Then redundancy of proteins will, after time, lessen as each cell realizes the other is producing it, and can be more efficient without. Gene transfer from one organism to another then occurs. As cell 2 replicates its genome, parts of it can end up actually moving in to cell 1's genome. And this transfer is actually naturally selected because anything that favors control for the host is better. So this movement of information from cell 2 to cell 1 allows cell 1 to have greater control over cell 2, and therefore this relationship continues to become more dependent. And eventually, cell 2 becomes wholly dependent on cell. So there is, if you think about it, when some of cell 2's genetic information moves into cell 1's genome, there will be a point where there's genetic information of the same in both. And so this intermediate stage is going to be present, but it does set the stage for mutations to occur and gene loss to occur, changing both genomes altogether. And after time, the host gains more control, and the endosymbiont loses autonomy. Over time, the single organism, almost all of the endosymbiont's genes are lost or transferred. So at what point does cell 2 do the two cells become one? Well, this change is actually gradual, and it doesn't happen at some sudden event. This was very important because it can result in genetic mergers that increase the complexity of the system, and thus has allowed eukaryotes to be so complex. Now, eukaryotes have actually ended up with chunks of multiple cells within their genomes. So in all organisms that are eukaryotes, there is some genetic piece from this cell 2 and some genetic piece from this cell 1 that have come together in our present-day genomes. And there is also actually some information from some other source that is believed to be another cell that must have come into this mix at some point in evolutionary history. And there's also a part of the genome that is unknown. And so that is a good question for the future, is how has some random information made its way into the genome? This could be mutation, it could also be something else. So now moving on here to chloroplasts for plants and algae. So as you may know, many eukaryotes just have mitochondria, animals and many other things. But chloroplasts have taken up another organelle that actually holds genetic information. And this is the chloroplast. So how could this have occurred? So chloroplasts come from cyanobacteria that is engulfed by the eukaryote. So this process one that I mentioned before has already occurred, and then cyanobacteria ends up coming in at some point later in evolutionary history. And this is where the beginning of plants and algae have come about. So now you can imagine the genomes of plants and algae also have this cyanobacteria component. So their present-day genomes do not just hold the two cells and some unknown information, and also another cell, like many animal cells do. They also contain cyanobacteria information. However, there are cases where horizontal transfer of chloroplasts have occurred. So as you can see, the take-up of a chloroplast has happened for one common ancestor and is sprouted off to many, many different types of plants and algae. But there are also cases where there has been a transfer of these chloroplasts into other organisms, and therefore they do not have a common ancestor to the organisms that originally took up chloroplasts. And so we actually know this because there are some organisms that actually contain chloroplasts that have two layers of membranes. And therefore we know that they are not genetically related to these other cells or organisms, but rather their chloroplasts have been horizontally translated. An example of this is Plasmodium. And it is a very lethal cell that we are actually capable of fighting, and with our new understanding of secondary chloroplasts, because we understand where it came from and how it works, and now we can target it with drugs and other methods to actually help other people.